Hello there. This is a presentation that was given at the International Cannabinoid Research Society in 2015 on July 1st. We are re-recording it just for better audio quality. And this is a presentation about a controversial topic related to cannabis speciation. There's been several suggestions of speciation in cannabis, whether sativa and indica are subspecies or their own species, and there's also a third one known as ruderalis. But in the last 10 years, an additional strain has emerged out of Australia, known as an Australian bastard sample, and we got access to the sample through the workshop in Seattle and decided to try to take an attempt at understanding the speciation event that's going on here, and if it's in fact a speciation event at all. So this is going to use both analytical techniques, what we'd call chemotype, and also genomic techniques, or genotype. So this is a chemotype and genotype analysis to understand a speciation, or putative speciation event. There's some conflicts that I'm an employee of Medicinal Genomics, which is a wholly owned sub of Courtagen Life Sciences. Courtagen focuses on sequencing children with epilepsy, autism, mito disease, and liposomal storage diseases, with a heavy focus on pharmacogenomics. And these are, of course, all diseases that we believe are centered around the ECS, or the endocannabinoid system. And Medicinal Genomics is the plant genomics side of the corporation. And we are looking to get better genetics and safety testing around the cannabis plant. So here's a picture of the plant. Again, this is a hybrid of a plant that was found in Australia. So it's been crossed with a purple skunk. The crossing, we believe, is difficult from the parental line, but once it's been crossed, it actually does appear to cross easily with other strains. It has not only a very bizarre leaf structure with phyllotaxy, but it also has some bizarre branching, and it's a little bit of a runt, we understand. Now, there has been a tendency in the cannabis field to classify things according to leaf structure, and we're going to draw some question to that here. You'll notice much of the taxonomy in the field talks about narrow leaf hemp, or narrow leaf drug type, or broadleaf hemp, broadleaf drug type, etc. But if you look at this plant, clearly it has different leaf patterns. If you look at the nearest cousin to cannabis, which is hops, it has the leaf pattern you see over here on the right. Yet you can find very distant species like Japanese maple that have a much more similar leaf structure than even the nearest cousin. So what's going on here? Well, we're beginning to think, are we really looking at eye color as a mechanism to discern Denisovans from Neanderthals? Is this really an accurate mechanism for taxonomy in the cannabis species? It certainly may co-segregate as an allele with indicas and sativas, but we don't think it's necessarily a very unique marker, and that particular marker may be a single nucleotide variant. Just like we see in the Hox cluster in humans, you can get single nucleotide variants in mammals and humans that can give very dysmorphic features, but that hardly qualifies as a speciation event. So we want to look at this differently. And the approach that the workshop jumped on was to, of course, use a microscope and see if they're trichomes. They found them. See if there's THC in theirs, and they can see THC peaks according to HPLC with UV confirmation. And they can also see some cannabichromene peaks. They're low in concentration, but that's not surprising based on Ernest Small's work showcasing that males tend to have less expression of these things. And the terpenes are there as well. You can see a handful of them compared to Cinex, which is a strain you can find in a dispensary in Seattle. But you can see myrcene in there. You can see linalool and beta-caryophyllene. These are all signature terpenes for cannabis, but they're by no means unique to cannabis. And again, the expression is fairly low. So this is certainly reassuring that yes, this cross is producing some of the chemotype we find in cannabis, and we don't necessarily know if that came from the Australian line or whether that came from the skunk line. It's quite possible it's from the skunk line. And so you then start looking at genetics, because in genetics we could possibly tease these two things apart. We could perhaps sequence them in a haploid-specific manner and separate the skunk haplotype from the ancestral haplotype. And so the first pass before going into such endeavor is just, do these things even have the Y chromosome? And there's a whole other presentation we've given at the ICRS in 2015 on how we develop this Y chromosome marker, but it in fact has it. It amplifies quite readily in triplicate, and it also has this SCCG region, which is quite surprising to us that we're getting amplification from this, only because the SCCG region has been we are targeting a region in the cannabinoid synthase pathway, and we've noted, and so has John McPartlin noted, that the cannabinoid synthase pathway is one of the most polymorphic regions of the plant. And so we had expected this to probably fail in that region, to potentially hit a new SNP we haven't found before, a very distant ancestral SNP, but that's not the case. It's amplifying quite well. And a lot of that hyperpolymorphism rate inside of the synthase genes is probably due to the selective pressure that's been applied with everyone breeding for that particular trait. So it's not surprising that that's there. It is surprising to us that we actually have some amplification going on there. But again, it could all be from just the skunk line. So how do we tease that apart? Well, we're going to go into whole genome sequencing into great detail here to suggest a way to actually tease that apart. So we queued this up with a Nextera system from Illumina, which is a library construction method that uses transposition to insert Illumina primer sites all throughout the genome. It's very effectively done with just 50 nanograms of DNA. This DNA creates a very broad array of DNA sizes. And if we just sequence those randomly, we don't really know the distances between the reads, and that's a bit messy. So what we tend to do is size select those things. So we know that every 250 base pair read we get, we know that its forward read and its reverse read are a precise distance apart, about 800, in this case actually it's looking more like 700 bases apart. That's incredibly important when it comes to mapping reads, and we're gonna touch on that. And then when you do map those reads to the genome, you can see over here on the left, a lot of these blue horizontal lines are paired reads that map, and they map with the expected distance they're supposed to map apart. In this case, that's 700 bases. The green and the reds are broken pairs, meaning only one of the read mapped, and that's perhaps not too surprising based on the way that we map this. If you map this just to the messenger RNA transcript, where it is precisely a 1.7 kb reference, well, the reads that land on the end are going to have a hard time anchoring their mate pair or their paired end to the actual reference, because the read will be half broken or only half aligned based on an edge effect of the reference. Now, this turns out to be an incredibly important point that we can't emphasize enough in dealing with polymorphic plants, because from this, we immediately assumed we had an alanine 5-threonine variant that is in the signal peptide, and maybe that is why this plant's not expressing as much THC. Although it looks homozygous here, we can see some heterozygous SNPs in the middle where things are paired, so this made us scratch our head a little bit. We then went into GenBank and realized many other people had seen this variant as well in the messenger RNAs that are out there. So it's not novel. It's been seen quite a bit before, and we wanted to dig into this a little bit further. If you take these sequencing reads and you map them just to these messenger RNA sequences, you get one answer, and that's an answer that is what we would call a reference bias. When you hand mappers or assemblers one hypothesis, they will force all the reads to try and fit that hypothesis. If you give the mappers, the mapping algorithms, the entire genome, it may find better homes for some of those reads and place them in different areas with higher mapping qualities. And we've shown that's exactly what's going on here. If you map to just the scaffold of 19603 in CanSat, you see this hyperpolymorphic picture down here, where all of these colored lines represent SNPs. If you map it to the entire CanSat reference, suddenly a lot of the things in the 5' end disappear. And even this particular SNP disappears. So this had us step back a little bit and look at some of the work published by Onofre and a little bit more scrutiny. And what we came, this is great work, by the way, that cloned a lot of RNA out of various plants. And by cloning them into bacteria and sequencing them with Sanger sequencing, they were able to get single molecules effectively cloned and phased. And what that means is they only sequence the maternal haplotype or the paternal haplotype in the process of cloning them into bacteria, so they could separate that. If you take a PCR product and sequence it, it will be a diploid representation or more of that region, and that's a very important detail for this particular project. However, nonetheless, this excellent work, there is some limitations to it, which is that the primer sites that they chose are actually internal to the gene. So the ATG up there in this primer site is actually the start codon of the gene. And this entire yellow region is the primer site. So all of that is synthetically derived sequence. It does not represent the sequence actually of the genome. It represents the sequence you gave the oligo manufacturer. And that happens to cover this particular SNP that we're interested in. So they actually can't see this. It's a blind spot in their method design. The result of this is that they may be selectively amplifying certain species and certain THC synthases as a result of using these primers. But nevertheless, it's a very good data set to look at to understand phase and variation. But we asked ourselves, all right, how do we do this with next generation sequencing? We don't want to be cloning all of these things into bacteria and picking colonies and sequencing these the old way. And it's a little bit on the edge of what the technology can actually do. This is a 1700 base pair gene, single exon gene. Sanger sequencing is known to give you accurate results out to maybe 800 bases. So you can get two 800 base pair reads to perhaps maybe 900 base pair reads to perhaps meet in the middle, and maybe where the reads overlap, you get some error correction in the overlap of the tail end of the Sanger reads, which can be very noisy. But you can also miss some of the sequence right there adjacent to the primer sites, and so the edges in the middle of this data are likely compromised, and we don't want any compromise because there's too much polymorphism in this interesting gene to have to worry about it. So what we decided to do is set up for a method that could phase the sequencing with the digital sequencing of next generation sequencing. And what we did is we take the amplicon. In this case, we're even using these Sanofi primers that we know have that limitation. But just as a proof of principle, can we then turn those into a shotgun library and sequence them? Now, the important part is when we turn them into a shotgun library, our reads on the Illumina systems are only paired 250 base per read, so they only stretch 500 bases in length. But if you make the molecules 850 bases apart, you can get paired reads of 250 bases that are separated by 850 bases. That's really handy because the start codon of the gene we can sequence with the forward read, and then the reverse read is going to land right in the middle of the gene, and we know it's going to land on some SNPs, and those SNPs are going to be a signature for what haplotype it's on. We can then align another read to that middle read in the middle of the gene and hop another 850 bases out to the end of the gene. And we will only do that in a very haplotype-specific manner. The SNPs that are in each of these reads we know are signatures for each haplotype, and we can do this technique which is known as haplotype walking, where when we land a forward read, we look at the reverse read, see what SNPs are there, and those SNPs are a signature for one allele. We then align a read that is paired, pointing even further into the gene, that matches those SNPs, and then we can construct the phase with Illumina data. Okay, so this is a little bit tricky to do, and we did do this across multiple different strains here, and we mapped them both to CanSat references and to our LA Confidential references, and you can see we more or less get similar results between the different cultivars that we're using. There's just some discrepancy between the references as to how many copies of the gene there are, and we're going to touch on that quite a bit with the work that we're doing next. Now, we do have to validate this. This is a new technique for phasing stuff with Illumina across 1,700 bases of highly polymorphic regions, and so the best way to validate this is to actually take these same amplicons and run them through another sequencing technology called Pacific Biosciences and use their circular consensus sequencing to derive a haplotype-specific answer. So this technology is a very clever tool, only typically has 85% accuracy. But they have a trick on how to get around that, which is to clone hairpins on either ends of your amplicon to make it into a circle. And when you do that, you effectively get a polymerase that starts to run around and sequence this in real time, but it goes around and around in a lap. And by going around and around in a lap 20 times, you can take the 85% random error and bring it up to 99.9% accuracy. And that's really critical. We need that accuracy, but we also need long reads. And those are two things that have been traditionally in sequencing orthogonal, but they're now beginning to be possible with these single molecule sequencers. And you can see down here, we get some beautiful results of doing 20 to 25 laps around this thing that drives the error rate way down. And so when we do this on ChemDog 91, we actually end up with 5 different genes. 5 distinct genes, about 99% identical, but one of them we can now tell has a stop codon in it and is probably a broken pseudogene. It is a C-terminal stop codon around 440, so maybe it's still functional. We need to do some functional assays to sort that out. But the other 4 copies, interestingly enough, there are no SNPs in the FAD binding domain. This is a region that Terpenes studied very well. If you have SNPs there, you get changing and folding rates in the enzyme. And there are no SNPs in the glycosylation sites, and there are no SNPs that meaningfully affect the signal peptide in any of these copies. Now, we now need to rethink about this cross. We now know that ChemDog 91, which is a skunk line, has 5 copies of the gene. So when this gets crossed with a Australian sample, which may have 0 or 1 copy of the gene, maybe 2, we don't know, what do we get? What's the independent assortment? Are all of those copies of the gene tandem in the genome? We don't know if they're on different chromosomes and how they're going to assort, but we can no longer assume that the Australian hybrid one is going to have just 2 genes, and then we sequence one of them and we sort out what's the skunk line, and the other one is therefore by default the ancestral Australian line. We don't know that. So what we have to do is run the Australian bastard amplicon on the Pacific Bioscience System, which is currently underway, to see what it looks like. How many copies does it have? Does it have 2? Does it have 5? And that should hopefully shed light on how this part of the genome has evolved in that process, and we may even be able to derive some phylotypic information from just the number of SNPs that are in the strain alone. Interestingly enough, there was over 114 SNPs in the Australian bastard THC synthase region. It's the most polymorphic thing we've seen yet in that region, and so the phasing information there should be very, very valuable and informative. Now, once we have these 5 haplotypes, we can then take our Illumina data that was done with this haplotype walking approach and map it against these 5 single molecule derived haplotypes and get beautifully aligned data with no SNPs. And that's what's expected to happen if the method's behaving. And what happens if you map it just to one of those haplotypes is you get this type of hyperpolymorphic alignment that you're seeing over here on the right. All these hashed colored marks are SNPs, and there's a whole host of them there, over 30, that are a result of collapsing all of that Illumina data to one specific haplotype and bringing out that 1% differences in the form of SNPs that aren't really SNPs. This is just compression into one hypothesis. So we've got a method of phase, and this is coming along quite nicely. But another thing that we pursued in parallel to answer the question of speciation was to not just look at this most interesting and highly selected region of the genome, but to go and look genome-wide. What happens if we sequence the entire genome of this thing? So we took 80 million reads from this, paired 251 base pair reads, and sequenced them, and then mapped them to 2 different references, and we in fact get different numbers there just because the LA Confidential and the CanSat references in fact have novel material between them. But if you map them back to this table over here is all CanSat based, so if you map them back to CanSat, we get 94.4% of the reads to map with the Australian bastard male sample. We then grabbed 2 other males that we have in the pipeline and saw that we got 99.6% mapping. So there's a big gap in the amount of data that can map from a male to CanSat. And that difference is likely the Y chromosome, and there's a whole other presentation we have on deriving the Y chromosome from Wi-Fi and from Grapesnapper. But if you take Grapesnapper, which is a male, and you map it with a different mapping algorithm, the numbers don't change very much, so the algorithms aren't playing a big role here. And if you take our LA Confidential data, which is a female, and you map it back to CanSat, you get a slightly different number, and the reason we're doing this is that this was actually sequenced with a different sequencer. It was we have some Illumina data on it, but we also have some 454 data on it, and it is also a little bit diverged, but that just shows you the range of a new sequencing technology on a different female, what happens. And then if you take Finola, which was sequenced by John Page's group as well, are done with a very similar sequencing technology as CanSat itself, Illumina data, and you map it back to CanSat, you get 97.7% mapping. And now that's a hemp line. You expect that. That should be a little bit more diverged. But to get a sense of speciation, take something like bonobo, which is another primate, and you map it back to human, you get 95.8% mapping. So this Australian bastard sample is clearly more diverged than bonobo is to human, and clearly more diverged than Finola hemp is to drug type cannabis. Now, these mapping numbers are, you have to take with a bit of a grain of salt. There are some things that can throw them off. For instance, if this Australian bastard sample happened to be heavily contaminated with a lot of bacteria, we'd get a lot of reads that didn't map. And so what you do to address that is you start, you normalize that effect and say, okay, give me only the reads where both of the reads map, forward and reverse. Those have to be from the organism because both of the reads map, and give me the number of them that are properly paired. How many of them are at the right distance apart? If you recall earlier, we have these libraries that are sized at 800 bases. And the reads should map such that both reads map and they are precisely that distance apart in the reference. And that properly percent paired is agnostic to any of the contamination that that we know can possibly exist in these types of metagenomic preps. And so what you see when you do this is you get 57% of the reads are properly paired. Now, what can break a pair? Well, the reads can be further apart, they can be closer than expected together, they can be inverted. All of these are signatures of structural variation, which are much stronger evidence for a speciation event. And as you go down this table, you can see, bonobo is 92.35% properly paired. So there's far less polymorphism and structural rearrangement between primates than there are between these plants. We did play around with hops a little bit, but hops is 3 times as large with the same chromosome N number, and that became a little bit more difficult to interpret. It's only mapping at 34% there. So what is kind of interesting is that 11% of the Wi-Fi and grape stomper contigs have no Australian bastard chromosome hits. These are basically the male-specific contigs that we've looked at. We tried to go through here and do some work finding male-specific contigs for the previous presentation about finding Y markers. And there's a handful of Y markers that are in the current male strains circulating in drug-type strains in the cannabis market that don't have any matches to this Australian bastard Y chromosome. So there's certainly some divergence there as well. Now, to the point of metagenomics and contamination, we do have a poster here using Pathogindicator, which is a product that we sell into the cannabis markets. The testing markets right now are now mandated to test for mold and bacteria on every pound or every 5 pounds of cannabis. And to do this type of testing, you can either use culture, which is only 1% of microbes culture, and it was invented 100 years ago, and there's all types of issues with what you can and cannot detect with those methods, and they tend to be very slow as well. And so we've moved all that stuff over to quantitative PCR. And in the case of quantitative PCR, it's very easy to design primers that specifically target Aspergillus, E. coli, and Salmonella, all these species-specific bugs people are worried about. But a handful of the regulations from the HP also talk about broad classes of microbes like yeast and mold, and enterobacteria and totobacteria. For those tests, we use 16S and 18S primers that when they give you an amplification, it's a metagenomic amplification. It's amplifying all of the species that are there with primers that are targeting regions that if you sequence those PCR products, you get a metagenomic profile that kind of gives you a digital read count assessment of the number of microbes that are present. And there's a great poster on this that goes through all the species we found on a couple dozen different cannabis microbiomes. But you can see there are, in fact, some mold that's growing on this plant, and so it's important to focus on those properly paired reads in this analysis because this can throw off some of those read mapping numbers. So the other aspect we took here was to not just look at read mapping similarities, but what if we can do a phylogenetic analysis of 100 strains and place Australian bastard on that map? What do we need to do that? Well, we have this process called strain-seq at Medicinal Genomics where we sequence about 1 million discrete loci in every strain, same loci in every strain. It's using a technique known as RAD-seq, which is a restriction enzyme digestion of the genome. And by doing this, what you effectively do is you anchor all of your reads next to restriction sites and you focus the read depth there. So it's kind of like getting high coverage readouts of the first page in every chapter of a book. And every time you approach a new book, you're always reading that first page of every chapter between the species, and so you can readily look for variation in the same place in the genome without having to do whole genome sequencing. Whole genome sequencing is great, but it can cost thousands of dollars to whole genome sequence cannabis strains, and we want something orders of magnitude less than that. So we designed this technique called strain-seq that with a simple size selection after digestion, gives us a couple million reads per strain and 100,000 variants per cultivar. And what you end up with is data like this. It piles up in very discrete regions, and depending on the read length you choose, you can find varying amounts of polymorphism that you can then use to build yourselves a nice phylogenetic tree. So this is a tree that was built off of 70,000 SNPs across about a 1.2 meg region in all of these strains. And what's really interesting about this phylogenetic tree, and the way to read this is the length of these vertical lines is a genetic distance. And so these hemp lines, USO31 and Finola, have the longest genetic distance away from everything else. But you can also get a sense of the relatedness to each of these strains by the distance on the horizontal axis. And what you can see is that many of the CBD strains are lining up next to the hemp strains. And that's actually been published as a source of where CBD may have come from. It seems to have more commonality with the hemp line. So Otto is a full 20% CBD strain. We've got CBD Mango Haze in here, an Australian Bastard snuck right between. This Pineberry sample we think is a sample swap. Sour Tsunami was done on the same plate, and that is more likely what would fall over there. We have all these hazes coming together. You can see all the Kushes coming together. We can see some relationships we know exist between Shokolope and LA Confidential OG Kush. We, of course, like seeing all of these repeat samples coming up side by side, Chem Dogs over and over again, Lucky Charms over and over again. And, of course, these two over here, these are 50-50 hybrid, 50% THC, 50% CBD strains that are sold in Seattle, and they're probably the same strain, just named differently, is our guess. And, of course, more replicates continue to give us confidence that the method's actually working. Now, there's many people working on doing phylogenetics in cannabis, and we're likely to all derive different maps. And so what we need is a mechanism to drive global consensus on these maps. And what we've been building toward that goal is what we call Canopedia. This is a Wikipedia that can allow the entire world to edit and augment information about these strains. The one thing that we don't believe should be augmented or edited is the genetics. And so what we're doing is building a tool that allows you to take the DNA sequence information and leverage the distributed consensus algorithms that are in the Bitcoin blockchain system to more or less drive consensus on genetics. So for those not familiar with Bitcoin, Bitcoin's this fascinating technology that has a cryptographic competition running on the most computer power in the world. Millions of computers now have more hashing power applied to the blockchain verification process than the top 500 supercomputers combined. Okay, so there is a monetary incentive for these computers to verify the veracity of a public ledger. And that public ledger right now is a transaction ledger, but there's no reason that can't be a genetic ledger. That we can use these tools to drive distributed consensus globally on what the cannabis phylogenetic tree is. So we've done that. We've basically made tools to submit 29 of these genomes that you saw in the last tree. Not all of them are up there yet, but 29 are now etched into the Bitcoin blockchain. The VCF file gets written. That's a variant file that has all the variants related to one strain that we found. And we basically make a QR code out of that transaction ID that sits in the Bitcoin blockchain. And that QR code will get you to the genetic information and to a timestamp upon which it was submitted. So we can get some type of digital notarization event of each strain and their genetics. And we believe this is the right way to drive a global consensus on a phylogenetic tree, because any one private database is likely to have a bias of its own and is likely to either be acquired or shut down or lose uptime, whereas when you get involved with distributed consensus tools, they are anti-fragile and that they are on millions of computers and no one can shut them down. And that we think is a very important attribute to an encyclopedia like this. So have a look at Canopedia. There's some really exciting work going on there. We're looking for collaborators and of course new hires to help us in the Bitcoin space. Okay, so this drives us to the last and final slide, which talks about, is this a new species? Well, Ernest Mayer's definition of speciation underscores the importance of separately evolving metapopulations. It specifically carves out the requirement for crossing, which is interesting and certainly controversial. So this is still, I'd say, a very bold statement to apply with the data that we have. I don't think we can call this a new species yet. I think what we need is the parents. And it's a harder problem than most people think. So if folks know or have access to the parents, please contact us. We'll sequence those and try to drive this to a final point of conclusion. And with that, I want to thank many of the members of the workshop who did all the chemotype work on this, some communication with John McPartland and Mark Emery, many many folks down at Florida that help with single molecule sequencing, the Medicinal Genomics team, but most importantly, all of this work was funded through Courtagen, which is this CLIA and CAP sequencing-based company looking at the genetics of cannabinoid response. And they're already finding some very interesting associations that have been published. One is this TRAP1 gene that's associated with chronic pain. We all have been hearing about chronic pain and cannabinoids. The biggest problem is subjectivity of diagnosis. Well, this here is a genotype that diagnoses 1 to 2% of the population with chronic pain, and we believe this particular variant would likely respond to a strong antioxidant like CBD. And then the other work they're doing is looking at CBD response in Dravet syndrome, which is a very interesting study, and they're starting to see some signals of the signatures, genetic signatures, that predict whether you will respond or not respond to CBD. And it's an important point in Dravet because there are populations of patients that get worse on CBD. It's not spoken about. There's certainly a lot of success with CBD, but about 10 to 15% of the patients get worse, and that's expensive. Seizures going up can be extraordinarily expensive, and so trying to figure out markers that can predict that can be very helpful for the field. So as you can see, it's very much a cannabinoid-centric group of people here, and we look forward to collaborations, and also I will throw out a shameless plug that we are hiring, and any folks who are interested in any of the work that we're doing here, please contact us so we can change the world here. Thanks.